Dude. So, good evening, students. Um, how all are you doing? I hope you're doing all good. So, last day of submitting is 20th of October. So, take your time. So, today is 30th October. So, we are on the 6th day. I hope you had your good lunch today, had good rest, and awake to go for the lectures for today. Besides what we are doing, if you have any questions, we can have a kind of little bit of discussion sessions. Otherwise, we can start with the sessions. If you have any other questions for science or anything else, I'm ready to take them, like a Q&A fire question type, and then we can start our session. So today I will not take you much time. That's the thing. It's they say it's easy to go for review, but when somebody starts to do it, somebody really gives their efforts. It takes some time, of course. In our time, we also took a lot of time, but yes, you can take some time if you want. But by then, you getting your certificates will be late. That's one thing. Yeah, as per your submission of your review, you will get your certificate. So I can share you with Ramneet ma'am. We had some free generals. So somebody wants to publish with us actually. We can put our name also, and we will help in rectifying it and publishing it. So in that order, we can do it in collaboration. Otherwise, if you want to do it on your own, you have to do it on your own then. So where can I share these? So these are free generals. So I shared them already with you on the. So if somebody is interested to publish on your own, please go ahead. Otherwise, if they need a little bit help from us to go reviewing, but in that case, go for free general or have a from university have access to free journals then. I mean, that's the thing. If you are submitting your review to us, um, so you can send us as a Word would be better, or PDF is fine to keep it as it is, or you can mail it to me and also to Edufabrica email address. At the time of certificates, you need to submit your details to the Edufabrica email address. In meantime, if you want to check your review and want to get published, there's a colleague of mine and me. We will go through your journals, and then we can tell you what things has to be cleared out. Then we can send it to the free journals that I have shared. So let me just share with her one thing she asked me today. Okay. So we will help in that your review is in order. So we have published quite papers before, and we can check them before you submit them. So don't worry about that. So is it fine if I share a little bit about electrophoresis and about my work also, what I did in my PhD, which we correlated to the technique and its outcome and its application. So last time when I was sharing, students get really bored, and I want to share like how you can bring the application of Western blotting into your real life work. So I will share one part of my PhD with all of you today. So I will share my story with you, how I get into PhD. So basically, I was a very, to be honest, a mediocre student. My story is very. I had luck. I had a very good luck. I have to admit. Um, I'm not that strong student from background, so I was getting around 70 and 75 percentage. Was not into the 90s ones. So most of the people who get to Germany or outside to good university, they have to be 90s part in their bachelors also, or to be gold medalist, something like that. So I did my average bachelor's with 67 percentage, and then I went for um, yeah, teaching job at the university, where I was teaching like all the bachelor student, BTech students, what I have learned for 5 months. But I had a thirst for more learning, so I went for my MSc to Germany, where I was interested with agrobiotechnology, agriculture plus biotechnology, to learn about more about transgenic crops. But while doing the course in the third semester, when I started to work practical in the plant field, I started to see that all the German people don't like these transgenic crops. They are not into so much, what do you say, they were burning our crops. Actually, whatever we were doing and experiments on the field, they come and they burn in the night. So that was very harsh feeling for me, and I was very hearted, disheartened of that situation. So I got a job in Germany at the Max Planck Institute. Before, in Germany you can do many jobs, so I was also cleaning utensils. They like organic crops. Yes, they do a lot organic crops. So they want to eat everything to be fine, not any have any GMOs or something. So I got a job at the Max Planck Institute as a helping student. So I was working with the histology department, where I was cutting microtome sections of lungs of cancerous and healthy lung samples of the mice. So they have one P2Y2 gene at the Max Planck. So they want to check what happened in the cancerous cells, lungs, and the normal one. So I was helping them in to get into the histology part. So I got interested into that field of cancerous field. So I got my master thesis there in the field of metastasis. So where I worked for 1 year, and based on that, while working in Germany in the Max Planck, I was earning also beside working as PhD. So I started to make money for myself and started to be self-dependent from the almost after 6 months or so. And my fees for Germany was nothing, it was zero. So it was almost, I was paying 400 euros for 6 semesters. The main expenditure was of my staying there, actually, of eating, and other everything was free for me. So it was all from the government paid agencies. So then I got, because of my studies, in my master thesis. It was very well established, very well accepted. I got an interview for my PhD at the hospital in the Göttingen, where most of the Nobel Prize winners are been granted. So it's a University of Wissenschaftler, they say. The city of Wissenschaftler means city of scientists. So they have a lot of physics, chemistry, biology, earth, solar system, human, mice, sheep. I mean, they are doing research on each and every aspect of life, that city. So so when I was staying there, so the PhD was a scholarship itself. So we were about to pay it around 1400 euros or so, 1400 to 1500 euros per month. So it's like 1.5 lakh to 2 lakh rupees per month of Indian rupees. So which was quite a lot for us. So my expenditure was mainly 300 euros for homestay, like house stay, the rent, and eating was 200 euros. So I for my masters, for going masters, I gave IELTS or TOEFL. I gave, so I cleared the exam at one go, academic TOEFL, and so yeah, so my expenditure was very less for PhD. Yeah, so to get the PhD, what you have to do is you have to start applying. There are some like Max Planck institutes. So mainly they will see your PhD master thesis. Max Planck Gesellschaft. So Germany is a huge. It's a huge place for research. So there is a biology and medicine. So there are lots of institute in. So you can check here, and then start looking for professors. So which professor will be best for you? There are also DAD scholarships. So you can also go during your PhD time for scholarship for some time with the help of DAD. And there are Fraunhofer institutes. So please look through these. These are the highest packed links there, so you can look for them. So before I go, I learned German language actually. So everything there is done in English. My whole PhD was in English. All the teachers, all professors, they speak English there. So that's not a problem. It's a multi-language nation now. But to stay there, you have to learn German. So before I went, I learned A1 basics. Then I go for it. So there are some IRTG programs. IRTGs, International Research Training Groups. So there you have to maybe apply on your own, and they will accept the people who are good for it. You can apply for it then. It depends. For the masters, yes, for sure. For PhD, it's not so necessary. But if some university could ask, it depends from university to university. Yeah, so I got my PhD actually just via an interview. I gave my presentation there, and without any, but I had to go through interviews, couple of interviews before I got selected. So my professor was Ali Al Moush, and he was a medical doctor, German based, and then he switched to research later on, and he was working with the phosphatases. So he's very well known in the field of phosphatases in the cardiovascular biology. So I got under him and I started to work with the redox regulation of PP1 and CTGF in cardiomyocytes. So I will be dealing with that part only. You applied in the Max Planck and they rejected your applications due to no proof of English. Um, that's not possible actually. English is not being a big deal there actually. They what they check is they check the student's mindset, how good you are scientifically good, how you can make things done scientifically done there. Bache, you have to Google these IRTG and internship programs on your own. Just Google PhD in your field, like cancer field, or it's in organic, or I don't know which microbiology, bioinstrumentation, biophysics and bioinstrumentations. Okay, just remain in the same field, bache. That will be, I will highly recommend. No, there is no age limit. They what they look for is just if students could able to handle 3 years, they could handle a project for 3 years on their own. So maybe you have to give them some IELTS score or something like that. So that might be the thing that was lacking. So there's a master internship for 6 months. Go for LinkedIn. A lot of students are present of our Indian-based uh, content. They went to Germany and they started to find uh, jobs. And some of my LinkedIn students, when they got admission in the masters in agrobiotechnology, and then they started to work in the Merck company, in the chemical biology company. So that's that. The best part is to after going there, there's a lot of options open for you. But from India, it's quite difficult because they have to pay for your visa, they have to pay for your coming, and it will be really expensive for you. Also, initially, you can't earn a lot being on student. So it's better. I will highly recommend those who are in the bachelor's go for masters as the moment, and then from the master, getting a PhD will be nice from Germany. No, it should not be from Goethe only. You can learn from anywhere, but Goethe is the best one. Their certificates are recognized everywhere. I also did from Goethe everything for my B1, zwei, B21. So that's the best. That's the best place to do so. Yeah. There is no age limit. So I started, so I had a quite a good experience of my PhD. So I was going through King's College London, the King's KCL, and then we had a collaboration with them. So we were going back and forth from there and here, and to exchange the data with each other. And I also got chance to go to a lot of conferences, international conferences in Italy, in Netherlands, in Germany itself. So that's you got really overview of the world while staying there. But in India, it's very um, you are very concise and very focused. But being an international, you meet a lot of international professors, so you get really good exposure to them from them, and they are really frank. To be honest, whatever you want to ask or go for a drink with them, have a chit chat, or go for a dinner with them in their house, they will not say anything no to that. So they are very, very open. They are very open for many things. So the best part of studying there is that everyone is working in your hierarchy. Yeah, Max Planck is known as the highest. They are the highest value of research. If somebody wants to have a career in R&D, then the Max Planck is there. But if you want to have professional experience, then University of Dundee would be better. Oh, MSc molecular biology. But is that Dundee in UK or in Ireland? University of Dundee is in Scotland. But the problem with this UK based countries is that Scotland, yeah, uh, that that after you're done with your studies, it's very hard to absorb in these countries. So many of my friends they did in UK. It's a long list of my friends, but they don't get the futuristic positions as they should get actually. Either you should do very high in your scoring, you should be very advanced in your there, but it's very hard to get absorbed later on. That's I saw in this UK, Ireland, Scotland, all these universities. So just be careful, ask around the students what was the future after you done your studies. So I did my PhD in almost 3 years actually. So I didn't spend 4 to 5 years. So almost in 3 years I finished. So I took almost 1 year to writing. So I was traveling around also Europe during that time. So it took me 1 year for writing. So it was finished in 3 years, my all experiments. So that's the best part. So you have to finish in 3 years, and also got my paper out of it. So a long gap would be a problem, but it's no problem. You can think for. So it's very difficult, bache, that you can get a 6 months internship from here. Yes, there are a lot of positions available, like uh, job vector for Germany. You can find lot of PhD positions, master's thesis positions, but they will prefer one who are living in Germany itself or in Europe somewhere. Not jobvector.com. So I'm just sharing whatever is coming to my mind. What is the exam name for PhD in Bark? What is Bark, bache? So in India, it takes some time. Even in Max Planck, if you do your PhD, it will take 5 to 6 years. So, I mean, no idea. I'm extremely sorry. I have very less knowledge about India. So I did everything there. Then I came back to India now. So I'm teaching at the moment part time. And yeah. So I try to, I'm making my career actually in via teaching itself. Just tell them for the gap period that you did something, things that you were looking for. You can say you may be traveling with your parents, with your family, or you had some genuine problem. Just share your things that were genuine things, like, so then there will be no problem, because Foreigners people, what they like is that being genuinity, being honesty. If you tell them the honest reason, then things will be much better for you. Nowadays, the UK work permit is easy as compared to before. You can get the UK visa quite easily. UK work permit visa as per the last years. So where students just have to finish their studies and they are told that come back to your own place. So now it's much better, but life getting adjusted in UK is somehow as different as compared to the Europe, to Germany, and even Sweden is also a good option if you have good money, because in Germany you don't have to pay the fees, but in the UK you might have to pay some fees then. Oh, sorry, in Sweden also. But Sweden getting a PR is also an easy task then later on. Just go through the website King's College London. There will be PhD applications, or you can go for master course at King's College London, then go for PhD later on. That will be better path actually. KCL. In my case, I was part of IRTG 1816. So you can look and apply there. Every year they are opening. You might see me also somewhere. So that was the place where I did my PhD. And this is the place I did my master's, Eustace Leavick University. So I will share these links with you. I would really recommend go for Germany. It's still students are still being invited. You can get very good chance there. It's just you need to have a focus planning how you can get there. Yeah, so for job you need a job search visa from India, JSV visa first. Then you can apply for job in Germany. Yeah, pandemic has made things. Funding is a big issue now. Postdoc, it's simple. Just apply with your whatever you are doing in your PhD. Just write to the professors. That's the best for you. Just write to your professors, like how you can apply there, how you could do it. So things are possible in that way. Sweden, I would say also. Denmark is also good. Germany. So all these three countries. Netherlands is also best. So all these European countries, I have seen the standard of college and research quality. It's very highly standard, and you get really all the equipments are present, and nothing like any fishy things will be happening to you. So that's the best part so far. They love it. It's a beneficial. Like somebody in their in the PhD, she did her defense in German, so she got postdoc very fast based on that. So it depends upon how you get it. It matters. Like till A2 level, A1, A2 level is good if you got it there, but not too hard level. They don't expect us, because German language is a difficult language, and they expect not us to learn everything in a single day. So if we are learning and trying to speak with them in a slow way, they really highly appreciate that. It's not compulsory. So they are around A1, A2, B1, B2, C1, C2, and within B1, B2 there are two groups. So 1, 2, 3, 4, 5, 6, 7, 8, 9, around 9 to 10 groups are there to finish it total. Oh yes, Utrecht University is the best one. I was there in that university. It's in Netherlands, right? Utrecht. Just go with it. Go with it. So if we have no further questions, shall we start? So I don't have much to teach you today. It's a very short topics for today. So day 5, day 6. Okay, up to which level we have to? If you can learn till A2 level, it's good enough. A2 level I would really highly suggest. At least 2 levels. So okay, I will start the session now. So welcome to the electrophoresis technique, where we'll be dealing with the various zonal and non-zonal techniques within the electrophoresis. So electrophoresis, what is electrophoresis? It's basically a physical methods of analysis which involves separation of compounds that are capable of acquiring the electric charge. So electrophoresis may be defined as a migration of charged particles through solutions under the influence of external electric field. Ions that are suspended between two electrodes tend to travel towards the electrodes that bear opposite charges. So there are two types of electrophoresis that we will be discussing today, that is zone electrophoresis and moving boundary electrophoresis. So zone electrophoresis has paper electrophoresis, gel electrophoresis, thin electrophoresis, and cellulose acetate electrophoresis. Whereas moving bonding will have capillary isotachphoresis, isoelectric focusing that we have discussed yesterday with the pH1, and the immunoelectrophoresis. So we will go through each one of them one by one, little by little. So zone electrophoresis. It involves the migration of charged particles on the sporting media. So we have some sporting media there like paper, cellulose, acetate membrane, starch, gel, polyacrylamide. So all these things are being used in that case. And then sporting media is saturated with buffer solution. A small volume is applied. On the application of potential difference at the end of the strip, we have redetermined the electrophoretic mobility. So they are quite good for the biochemical investigations. Cost is low, easy maintenance, and quality of sample can be analyzed with the help of that. Disadvantages are unsuitable for accurate movability, isoelectric point determinations, and due to the presence of sporting medium. and technical complications such as capillary flow, electroosmosis, absorption, and molecular savings are introduced. So general method of operation is that you have a saturations of media with buffer, then you add a sample application, electrophoretic separation is done, then removal of a sporting media. So in the instrumentations, you can see electrophoretic chamber, electrodes, diffusion barriers, sporting stabilizing media is required. So this is the paper electrophoresis looks like. So you have a DC battery with 45 volt, and then you give your positive charge and negative charge like this in the electrolyte. Then this is your paper filter based on these two glass plates, and this is a pencil line being put up there. So this is the apparatus for your paper electrophoresis. So this could be of two type, that is horizontal paper electrophoresis and vertical paper electrophoresis. In the horizontal, so you have first you have electrode positive, electrode negative, then you have a airtight housing. Same in the vertical, you have positive and negative and air house, tighting house. So in the horizontal you have filter paper strips like this maintained in the straight way, horizontal way, and inside is your buffer solution. Whereas in the vertical you have a sample application point from the top and a filter strip is like put up there like this. So that's the main difference between your horizontal and paper electrophoresis. So in the filter paper we use Wattman paper number 1 and 3 of 3 millimeter in strip of 3 to 5 centimeter wide, which has a good effects, and separations takes place around 12 to 14 hours. It is economical, easy to use, but there are some disadvantages like electroosmosis. Certain compounds like proteins, hydrophilic molecules cannot be resolved due to the absorption and ionogenic properties of paper, which resulting in the tailing and distortion of the component bands. So there's some problems with the bands coming out because of the ionogenic properties and absorptive properties of the paper. So these techniques are highly what so far in these techniques, so we will discuss each one of them, but most of the time your paper gel electrophoresis is used mainly, and capillary electrophoresis is used sometimes. Sometimes isoelectric focusing is also in use, but rest of the one are not so much in demand and not highly used in the lab actually. So mainly SDS-PAGE is used with the denaturing and naturing agent. These two we will be discussing, and some I will play one lady is explaining about SDS-PAGE and gel electrophoresis very nicely. I wanted to create one presentation for you, but I think I will play that video in so that we can save some time and understand things better. So this is a continuous electrophoresis. Here you used for the preparative scale purpose, and sample volume of 0 to 2 ml per minute has been valve device is applied to it, and potential difference of 500 volt is applied. And here you can see that there is a positive and negative electrode. You have a sample reservoir here, and then you have sample components. These are your filter papers, and then being dissolved like this. So they are coming in this form, and then you can have your samples to be cutting up like this, as being separated like this. So gel electrophoresis is brought upon through the sieve technique. So mainly gels are made up of act as a molecular sieve. So gel is a colloidal in the solid form, 99% in water, and it is important that support media is electrically neutral, and different types of gels can be used: agar, agarose gels, starch, cephadex, polyacrylamide gels. So as per the gels are, so you will have different applications of your gel electrophoresis. And porous gel act as a sieve by retarding or in some cases completely obstructing the movement of macromolecules while allowing the smaller molecules to migrate clearly. So smaller molecules go down very fast and the larger molecules they remain up. So this is a casting tray for making gel slab. Then you put agro solutions into the casting tray. Then you comb with the help of comb. You make a well, some well in it. and then you put your samples into it, and then after putting the samples you attach to the power supply with a positive and negative electrode, and hence your electrophoresis chamber will start. So this is a basic principle that is being used in the gel electrophoresis. And then gel electrophoresis could carry out in two types, two methods: vertical starch gel electrophoresis and horizontal. So what kind of gels you could use? A small difference between agar and agarose gel. So agar is a mixture of polysaccharide extracted from the seaweeds, whereas agarose is a purified uncharged polysaccharide derived from the agar. So agarose is chemically discharged repeating units of 3,6 anhydride L-galactose. And agarose gels are fragile. They are actually hydrocolloids, and they are held together by the formation of weak hydrogen and hydrophobic bonds. So these are various advantages. They're easy to prepare, resolution is much better, large quantities of proteins can be separated, sharp zones are obtained due to less absorptions. So electroosmosis is high in this case. One of the disadvantages, resolution is less as compared to polyacrylamide gels. So that's why whenever you're doing your electrophoresis, so I always prefer to go for SDS-PAGE, in which in your polyacrylamide gel you add SDS into that become your SDS-PAGE at the end. So this application of gel electrophoresis that we have discussed is used for the immunoelectrophoresis. So in the solid state it looks like this, but when you make temperature of 45 degrees Celsius, initially gel looks like this, then you make some aging, after sometimes your polyacrylamide gel looks like this final gel structure. And again if you make it to 100 degrees Celsius, they broke it, and then again at 100 degrees Celsius they become in the solid state. So let's play one video about agarose gel electrophoresis.
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the samples spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on. Then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
Go somewhere. So I hope it was clear with the gel electrophoresis, how you put your samples, how gel runs, and how your DNA material moves from negative to positive side, and whether it's a protein or a gel. So they always tend to move from negative to positive sides with the time. So that's the basic principle behind that. And then there's a polyacrylamide gel electrophoresis. It is a prepared by polymerizing alkyl amide monomers in the presence of methylene bisacrylamide to cross-link the monomers. So your acrylamide, so this is the structure of acrylamide. They make a polyacrylamide with the help of methylene bisacrylamide actually, and they make a cross-link linked lectures like this. So this has become a gel-like structure like this at the end. So they are held together by the covalent structures, and they are separated on the basis of charge to mass ratio and molecular size, and phenomena called molecular sieving. And gels are quite stable over a wide range of pH and temperature in this case, and gels are different pore size can be formed, and samples can be separations is good comparatively. Just a second, I have to send someone something. Forward. So there are types of pages. There are two types of page, that is native and denatured. In the native, your structure of a protein remain intact as how it is, and its separation is based upon the charge type and charge size and shape actually, and usefully for the separation for the purification of mixture of proteins. And this was highly original mode of electrophoresis. And separation is based upon the molecular weight of proteins, and the commonly used method for determining the molecular weight of proteins in that case. So page procedure, to know about the page procedure, let me take you to this SDS-PAGE. So maybe I can play this video for you for next 13 minutes. So where your SDS-PAGE and all these things will be cleared off very nicely. I wanted to create this presentation for you because I found it to be very accurate, but due to time constraint I couldn't. So I'm playing this video by that lady. She explained this data very nicely. So please go through this and maybe try to make notes out of it.
Guys, in this video I'm going to speak about SDS-PAGE preparation, the discontinuous gel system, and the separation principle. So at the end of this video you will get a very good idea about SDS-PAGE. You will understand everything about the principle and the technique. So I'll start first talking about what is SDS-PAGE. SDS-PAGE is an electrophoresis technique. It's used for protein separation. And the gel used for SDS is polyacrylamide gel. Acrylamide is an inert material, so it doesn't interact with the proteins, which is a good feature, and it forms a matrix. The pore size of the matrix can be determined by the concentration of the polyacrylamide gel, so it's a good gel. The SDS gel is composed of two layers of the gel. The first is the stacking gel. The second is the separating gel. In the stacking gel, we have the wells in which we apply the protein samples, like here. And then we apply an electrical field to the gel, going from negative on the top to positive on the bottom. The electrical field is applied by a battery. And when the electrical field is applied, the proteins start to migrate from negative to positive, and they are separated in the separating gel according to their, like this, according to their molecular weight. This is the general description, but I'm going to speak about everything in details. First of all, to understand the principle of the SDS-PAGE, we should speak about the sample preparation. In the sample, we have the proteins, and we also have what we call the denaturation buffer, or the sample buffer, or the Lamley buffer, which is composed of several substances or materials. And the most important two materials of this is SDS and the beta-mercaptoethanol. So I'm going to speak about them first. SDS is an anionic detergent. It has a polar side and a non-polar side, a polar side and a non-polar side. When SDS is dissolved, it gives a negative ions, and these SDS ions bind to the proteins on the side chain of the amino acids that in the protein. So SDS gives the protein a negative charge. Whatever is the original charge of the protein, even if the protein is originally negative or positive or neutral, the SDS gives the protein negative charge. The SDS also denaturates the protein. So it converts the protein from its tertiary and secondary structure to its primary structure by breaking the bonds that are implicated in the formation of the tertiary and secondary structure. Similarly, beta-mercaptoethanol also has a role in the denaturation of the protein. It's a reducing agent. It reduces the disulfide bond in the protein, so it separates the subunits of a certain protein. So this is the protein, this is the amino acid or the peptide chain of the protein, and then the secondary and tertiary and quaternary structures are formed by non-covalent bonds like the ionic bond, the hydrophobic interaction like van der Waals bonds, the hydrogen bonds, and the disulfide linkage. And these bonds are broken by the SDS and the beta-mercaptoethanol. At the end, we will have the protein as a peptide chain. So the shape of the protein doesn't play a role in the SDS, and also the charge of the protein. So neither the shape nor the charge of the protein plays a role in the SDS-PAGE, only the molecular weight of the protein. So this is SDS and beta-mercaptoethanol. We also have the tris hydrochloride, which is a buffer that is of pH 6.8. It also gives the chloride ions. The chloride ions are also important in the mechanism of SDS. I'm going to speak about them. Bromoethanol blue is a staining dye. It's used to give the sample a blue color, so we can detect the sample on the gel, the level of the sample on the gel. Glycerol increases slightly the viscosity of the sample. And also we use distilled water to continue the volume, and we use distilled water because we don't need ions in the sample. Now I'm going to speak about the discontinuous gel system. So we have two layers of the gel, as I said previously, the stacking gel and the separating gel. And they have different pH. So the stacking gel has a pH of 6.8. The separating gel has a pH of 8.8. We also have the two electrodes that have pH of 8.3. There is something called the migration buffer, and the migration buffer contains tris and the glycine. Glycine is also very important in SDS-PAGE, so I'm going to speak about glycine. Glycine is an amino acid, and it's the simplest amino acid because the side chain is only a hydrogen, and it says vitorion. What does that mean? It means that in acidic pH, so below 2.3, this amino acid is protonated, so it's positively charged. And in basic pH, over 9.6, it's negatively charged because it's deprotonated. Between these two values, which we call pKa1 and pKa2, this amino acid is neutral. And it's totally and exactly neutral in the very middle point between pKa1 and pKa2. So how can we know it? We take the 2.3 plus 9.6 divided by 2. So we get the value of 5.95, which we call the isoelectric point. In the isoelectric point, the molecule is exactly neutral. Above this point, so at pH, let's say pH 6 or pH 6.5, the proteins start to be slightly negative, and the negativity of the protein increases gradually between 5.95 and 9.6. So we should know that in this area it's slightly negative, and the negativity increases. So let's see. When the pH is in the electrode, the pH is 8.3, and then glycine is negatively charged. As you see here, glycine here at 8.3 is negatively charged. So the glycine will migrate from the negative towards the positive pole because it's negatively charged. But when it enters the stacking gel, in which the pH is 6.8, the negativity of the glycine decreases, and then the mobility of the glycine becomes so slow. So I will speak about each one of these separately. So let's first take the stacking gel. In the stacking gel, the pH is 6.8, and here we have the sample. So in the sample, we have chloride ions. As I told you, we have the tris hydrochloride, which gives chloride ions. We have the protein SDS complex, which is negative. Chloride is also negative. And we have the glycine, which is at pH 6.8, neutral to slightly negative. So they migrate in this order. So first we have the chloride, then we have the protein, then the slowest is the glycine. So as you see here, the proteins are stacked between the chloride and the glycine. And because of this, we call this the stacking gel, because the proteins are stacked between these two. And when the three molecules reach this line, they start the separating gel. In the separating gel, the pH is 8.8. This is the separating gel, and the pH is 8.8. So the migration takes place in this order. So first we have the chloride, which migrates so fast toward the positive charge. Then we have the glycine, because when the pH is 8.8, the glycine is much more negative, and so it becomes faster than the proteins. And then we have the proteins. So you see here the proteins are left free behind. So the proteins are free behind, there is nothing, and then they are separated freely in the gel. The question is, why do we have this discontinuous gel system? And the answer is, when we apply the sample in the well, the sample is about 3 or 5 millimeters. And then the proteins will not be applied to the gel at the same time. So if the separating gel starts from here, the proteins will be applied to the gel not at the same time. But when the proteins are in the stacking gel, they are stacked together and then they enter to the separating gel at the same time. And they are separated to give the bands, especially like when proteins have the same molecular weight, they should migrate together at the same time to give this band. Now the separation takes place because this gel is a matrix, as I said before. So you can imagine it like this. This is the polyacrylamide matrix. This is the sample. So the sample migrates from negative to positive. And when it starts in the separating gel, the large proteins cannot migrate farther because they stick in the gel earlier. I should say that the stacking gel is a very low concentration of polyacrylamide, and because of this, the proteins do not stick in the stacking gel because it's so diluted. It's not concentrated, so the pore size is so big. But in the separating gel, the separating gel is more concentrated, and so the proteins will stick at a certain point. So the larger proteins stick earlier, but the smaller proteins migrate and stick later. And then we will get bands, different bands of different sizes of the proteins. This was everything I wanted to tell you about SDS-PAGE. I hope you enjoyed this video and you benefit from it. If you have any questions, you can leave your questions.
So students, I hope this was clear.
Hello guys, in this video I'm going to speak.
Sorry, no more videos. So that was.
Hello guys, in this video I'm going to.
It happens sometimes. So that was about SDS-PAGE, mainly the principle like chlorine, glycine, and your proteins. How they were first like 1, 2, 3, then it's 1, 3, 2. How proteins has been exchanged in the stacked gels with the 6.8 and 8.8 pH, and the stacking and the separating gel. So how the different pHs have the different roles. So that's was the main idea behind that, and why they stuck at a particular level, because of the polyacrylamide gels have these sieves, and the higher molecular weights they stick at the top, and the lower they stick at the bottom. Okay, I hope this was clear for you today, the SDS-PAGE. So that's something about the text. Just go through these texts, long text. So you have heat, reductant, and SDS, denatured proteins with bound. So we know that SDS mainly breaks your covalent bonds, whereas beta-methyl breaks the disulfide bridges. So you have a denatured sampling of a sodium disulfide. SDS is an ionic detergent that binds to the stoichiometric number of polypeptide bonds separating on the basis of the molecular weight. So the basis on the mixture of proteins and cross-link, then you stain to visualize them. And further, a suspension of granular starch should be boiled in buffer to give a clear colloidal suspension. A suspension on cooling set as a semi-solid gel due to the intervening branch changes of myelopectin. In order to avoid swelling and shrinking of petroleum jelly is used. So we have discussed this. And there is a thing called pulse field gel electrophoresis. It's a gel electrophoresis method, which is a technique used for the separation of large DNA molecules by applying to a gel matrix an electric field that is periodically change directions. However, the periodic changes of the field directions of the various length of DNA led to the change of the different rates in this case. So the voltage is periodically switched among three directions. So in this case, we change the voltage into three directions. One that runs through the central axis and then run at the angle at 60° either side. So one at the central and one at the either sides. The pulse times are equal for each direction, resulting in net forward migration of the DNA. So one is on this direction we run, and then one run on this direction, and one we run on this direction. So that way your DNA is separated by pulse generated electrophoresis. Then comes your next part is as a thin layer electrophoresis. So in this case, studies can be carried out in the thin layer of silica or alumina. So less time consuming, good resolution is possible in this case, and widely used in combined electrophoretic chromatography studies in two-dimensional studies of proteins and nucleic acids hydrosylates. Comes the last case, in this case cellulose acetate electrophoresis. It contains 2 to 3 acetyl groups per glucose unit, glucose unit, and absorption capacity is less than that of paper, and it gives sharper bands, provide a good background for staining glycoproteins. Its applications are widely used in the clinical and biological protein samples like albumins and globulins, and this is an alternative to the paper electrophoresis. Then comes the moving bound electrophoresis. Moving bound electrophoresis that allows the charged species to migrate in a free moving solutions without supporting the medium. So in this, instrumentation used is U-shaped glass cells of rectangular cross sections with electrodes placed on one of the each limbs of the cells. Sample solution is introduced at the bottom of through the side arms, and the apparatus is placed in constant temperature at about 40 degrees Celsius. So here you have moving bound electrophoresis. You have positive and negatives. This is very old method actually, moving bound electrophoresis. So you have ascending boundary, initial boundaries here. So you have descending boundaries on this side, and in between you have protein molecules. So in this case, you can see observed boundaries of optical systems. There are some boundaries being seen here, and these are its advantages. It is used for the macromolecular systems analysis of biological mixtures. Then comes the capillary electrophoresis. This is a principle behind the electrophoresis. Charged molecules will migrate towards the opposite poles. So charged molecules will move towards the opposite poles, separate from each other based on the physical characteristics. Capillary electrophoresis has grown to become a collection of range of separation techniques which involve the application of high voltage across buffer-filled capillaries to achieve separations. So in this technique, by performing the electrophoresis in buffer-filled narrow bore capillaries, normal from 25 to 100 millimeter internal diameter. A high voltage is applied. Capillaries are typically 50 micrometer in a diameter and 0.1 to 1 millimeter in length. Due to electroosmotic flow, all sample components migrate towards the negative electrodes. So this is the some method to show you that you have a cathode and anode. You have your all charged particles to be arranged at cathode first. Then in capillary electrophoresis you started. Then there is a light source here. Then this is all in buffer. Then which is attached to this light source with the photodiode to the computer, where you can see the output of your capillary results. Then comes the electroosmotic flow. This is the surface of the silicate glass capillary contains negatively charged functional groups that attract positively charged counter ions. The positively charged ions migrate towards the negative electrode and carry solvent molecules in the same directions. So positively charged ions move faster and negatively charged ions moves slower in this case. So here you have a buffer electrolyte, and then these are your samples, positive and negative. Sample inlet is done, then there's a capillary tube inside this, and here you can have the data acquisitions of your electrophoresis. Then comes the isotachphoresis. The technique of isotachophoresis depends upon the development of potential gradient. Based on the principle of moving boundary of electrophoresis, a leading electrolyte with a higher mobility than the analytes and a trailing electrolyte with a lower mobility are used. Where the separation of isoelectric focusing depends upon the existence of the pH gradient in the system, and the technique of isotachophoresis depends upon the development of the potential gradient. In this example, we can see that there are three particle classes with different charges are being separated and pre-concentrated by the electrophoresis. After the separation is concluded, all the particles move at constant speed, and this is known as your isotachophoresis. Then comes your immunoelectrophoresis. In this case, antibodies are produced by immune system in response to the foreign macromolecules. And each antibody binds specifically to one features on one macromolecule. This allows the use of antibodies for detection and quantitation of specific proteins in the complex mixture. So when electric potential is applied to the study of antigen-antibody reaction, it is called immunoelectrophoresis. So here we are checking the how antigen and antibody they are reacting together and checking their interaction in this case. The antibody are electrophoretically separated and antigens diffuse towards each other, resulting in the precipitin arcs where antigens antibody complex forms. And this technique has been referred to as immunoelectrophoresis. And this electrophoresis, as a result, precipitin arcs will be formed due to the antibody antigen complex formations. So, yeah. Then comes this we have done in details yesterday, isoelectric focusing, how they are being changed, charged from minus to positive, and they come at the center point of the isoelectric focusing point, and then we wash them away. This we also did yesterday in detailed manner, 2D gel electrophoresis by that professor video, and so I will not like to repeat it. So thank you very much. So here we can see a paramecium parlor where three ladies they came, and this is guide they're in the Halloween party, and they're in the great costume. So this is a restriction enzyme actually, who coming to be part of the party. So this is a funny story. So I think I will stop it here. Tomorrow we will deal with the various other parts of it. So I can take your questions now. So I'm also sharing one protocol for gel electrophoresis for DNA. So, yeah, I want to share with you a little bit more work actually. Electroblaster fertilizers, which techniques? Oh, I have to Google this about that. This is a disease. So we want to check how to check this diagnosis. So erythroblast fertilizers. So it could be done by routine blood test. By checking the blood test, you can do it. No, it's a blood test book, not with that. It's a blood group testing. So if you have still time, can we go through my presentations of my PhD, or we just stop it here? We can share it tomorrow also. What do you say? Because tomorrow we also doing blotting technique. It's up to you how you want to do it. Okay, I continue. That was my defense thesis. That was my first project. I will not share this now. So I will share this part with you. So something about ER stress signaling pathway. So ER stress signaling pathways is done actually because of the three proteins, that is ATF6, RE1 alpha and PERK. So whenever they are dimerized or phosphorylated, so there is a BIP proteins. It is initially attached to it, but when it is removed, they become dimerized and they get phosphorylated both. Whenever they are done, this PERK protein or RE1 alpha, the first one PERK protein will do the kinase activity. With the kinase activity, it will with the presence of EF2 alpha phosphorylation will happen, and then leading to the either stoppage of translation, there's a break, or there's a production of ATF4, and then it will produce CHOP4 protein. In the presence of CHOP4 protein, it will do the nucleus transcription, folding enzymes, and upregulation. Then further, ER1 alpha, it will do the RNA splicing of XBP1 mRNA, leading to the XBP1S mRNA production, leading to the translation of XBPS1, and then leading to the functions of nucleus. Further, this was because of the RE1 alpha when it get dimerized. Then there is ATF6 also present, which get migrated to the Golgi body, where the S1P, S2P parts are broken out and they are cleaved out. Then there is a cleaved ATF6, and this cleaved ATF6 goes to the nucleus to do further functions. So we want to check in my thesis that whether the CTGF part, the connective tissue growth factor, which is this one, which is a folding protein, is having any play role with the ER stress, or it is giving ER stress to the proteins, or ER stress is giving to that protein. So we want to check vice versa relationship. So we use mainly in these techniques Western blot techniques, that is SDS-PAGEs. This is the structure of that protein, which has in four compositions: IGF alpha, TGF beta, VGF, and VGF. And these each components are combined together by the various disulfide pitches. So in detail, we want to check. So this is how you have to present your master thesis whenever you have your PhD thesis. You show your first hypothesis, what you are looking for. Tell about each structure, that what is the structure is. Then you come to your hypothesis, and then I try to make a figures like this. So this was my style of presenting there. So test the hypothesis whether redox and ER stress affects the CTGF and vice versa. In details, following questions were addressed. Is CTGF affected by the redox ER stress in cardiomyocytes? And is ER stress and UPR pathway affected by the expression of CTGF? We attempted to answer the above questions by using a combination of non-reducing gels and incubation of cells with the redox ER stress eliciting agents. Furthermore, we also use siRNA technique to knock down CTGF in cardiomyocytes and assess the impact on the various ER stress markers. So here you can see that protein expression of CTGF in end stage human heart failure. So first we want to check what happened to CTGF and the end stage heart failure. So these are non-failing samples. ischemic cardiomyopathy, dilated cardiomyopathy. So these are both disease state cardiomyocytes, and the expression of CTGF as compared and tubulin we has taken as a control, which is present equal in the all amount. So we compare with them, and what we saw that the non-failing as compared to DCM, it is highly significantly up. So that means that the CTGF is quite important in the heart failure. So CTGF is a cysteine rich protein dysregulated in the heart disease. Then we check the effect of CTGF proteins exposed to hydrogen peroxide and DDT. So we did some time and concentration experiments. So this is a time dependent experiment, and this is a concentration dependent experiments. So here you can see the time dependent experiments with the H2O2. We have 100 micromolar of H2O2 and 100 micromolar of H2O2. Here time period window is for 30 minutes, and here the time period is minute for 6 hours. Here 6 hours, here for 30 minutes with a range of 3 minutes up, and here it's like 3, 6, 10, 15, 30, and here I took 2 hours of range, 0 hours, 2 hours, 4 hours, 6 hours, and then you compare with the control and then check what happened with these conditions. So you saw that there's a bit increase in concentration of H2O2 of CTGF with increase in H2O2, but then they start to decrease in first 6 minutes. And also in the case of 4 hours we saw a bit higher, then it started to decrease. So we don't know the reason behind it, but something H2O2 is doing to the CTGF for sure. Then we check the activity, the gels of in non-reducing conditions and reducing conditions. So these are your non-reducing conditions. So these are our non-reducing conditions. So where we have no DDT in our sample, and these are reducing conditions. So two kinds of gels I'm running, non-reducing and reducing gels, in the presence of 50 millimolar DDT with a time span of this much. So here you can see is that there is a very nice skill bar is open. That means there is some disulfide bridges are happening for sure. So that's why we see these things. So also we saw CTGF to be present in the concentration dependent of DDT 50 and 100, that there is a dependent, there is some disulfide bridges are happening. So you should not go in details about them. What I'm saying is just you have a gist about how you do your experiments, how you maintain your control, how you see your pharmacological agents. Like in this case, I did some experiments with thapsargine, tunicamycin, and tunicamycin. So I check how the expression is increased in this case with CTGF. So just want to let you know how things are done. So you should not think in detail. So I did all these experiments to check of CTGF with various agents, and then I check to check with the XBP1 how XBP1 being spliced in the controlled and SI CTGFs, that is knockdown CTGF. So I knocked down the CTGF in cardiomyocytes at mRNA level, and then we saw that CTGF is knocked down very highly level, and also the XBP1 was spliced. We can see the spliced version of it. So that proves that ER stress has role with the CTGF. Then we also checked then by the Western blotting. Here you can see in the Western blotting you have a decrease in the expression of CTGF, and also we saw decrease in other expressions of markers of a ES stress marker. So that means CTGF is correlated with all the CS stress markers. And also we saw the ATF6, which is present outside in the control, but in the knockdown it's present in the nucleus. So that means also there is a ES stress is happening whenever there is a knockdown of CTGF protein. So they are interconnected with each other. So this is a summary of all these factors on the CTGF agents. That is, there is no effect of BFA, MAG, pH on the CTGF, but we saw difference on the oxidizing agents, ER stressing agents, ES stress markers, heat shock, HSP47, end stage failure. So we can say CTGF data argue for interconnection of CTGF and ES stress R, ER stress, and ER stress modulates CTGF and vice versa. So that's it. So that's work. This work is not published, but my previous work is published in Scientific Reports, Nature, with my name there, Simranjit Singh. So yeah. So this was my paper. Thank you to all people. Thank you very much. I will close it now. So thank you, students. I hope everything is clear for today. We can leave now for as of now, and then we see us same time, same place, with more details about electrophoresis, about southern blotting, northern blotting, and western blotting. So we will deal with that part tomorrow. For today is so if you need some time, you can take some time. No worries. If you are in need of time, just take your time. Then we can see it how it is. But most of my students, they have sent me within that specific time. So yes, there are a lot of, in my during my time, I had a lot of positions I've seen for 6 months, and they're still coming up. You can check at the jobvector.com and then look for it. Okay guys, I have to leave now for my next session. Yes, you can submit to me. No worries. I hope your all questions are clear. Otherwise, you can write me. I will try my best to answer them. Okay, student. Bye. Take care. See you tomorrow. Did I share already the file for today? Electrophoresis. Yes. Bye bye. Take care.